welcome to this week's IDFP seminar. Our speaker today is Dr. Gülregin Yochai. She's a professor at the Faculty of Medicine at Tel Aviv University, and she's also the director of the Sheba Pandemic Research Institute. Her research has on COVID-19, vaccine effectiveness, correlates of protection, and the immune response has led to major policy decisions, and we're very excited to have you here today, and I'm looking forward to your talk.
Thank you so much, and thank you for inviting me and coming back here. Always feels like coming back home, so thank you. So I'll try to tell you today a story. Actually, it's a story of two tales, which might seem really unconnected, but I hope I'll manage to make the connection. One is actually talking about pretty much 2 years, maybe a bit more, of research we've been doing on COVID and how that led to the establishment of SPRI, which is the Sheba Pandemic Preparedness Research Institute. And the second part will be on our initial achievements since then. And it's sort of a shift in the story, but you'll see. So let's start. So everything started to all of us, I guess, back there in April 2020 is when we had the first cases of COVID. And at that time, we started an initial study on the immune response to SARS-CoV. And but when the big thing happened was on December, actually I remember it, December 19th, it was Saturday, vaccine was available, and we started vaccinating healthcare workers at our hospital at Sheba. On a Saturday weekend, people came, stood in line, and we understood at that point that we are the largest tertiary medical center in Israel. We have over 10,000 healthcare workers. We knew that we're going to have a successful campaign and rollout, and this is the right place to do a follow-up study and look at real-world vaccine effectiveness, both on symptomatic, asymptomatic infections, infectivity, and study the immune response, not just to the infections, but also to vaccine, and maybe we can find correlates of protection. So I went to the CEO and I said, can we recruit, maybe 1,000 of the healthcare workers? And he said, by 1,000, recruit everybody. Whoever agrees, recruit them. And he really was very supportive in giving the budget and all of that. And that is how actually our big healthcare worker cohort started. So we had at that point already vaccination data and PCR data was collected for all of the healthcare workers because we were already nearly one year in the pandemic. We had detection of symptomatic cases, and every morning each healthcare worker would get a text message saying, do you or your family members suffer from ILI today? If they said yes, they had to go and do a PCR testing. And everybody was at that stage, again, very compliant to all of this. And we had a computer-based tracing system, so we could detect any exposure, or at least most, obviously not any, but most of the exposures, also the asymptomatic cases, because when either a healthcare worker said that his family member had ILI, or if we knew that we had an infection, a COVID infection in the hospital, we would immediately screen all of the healthcare workers who were at some contact with them, and that way we really had a lot of data. And as we got the permission, we ran very rapidly this study, additional part of the study, and had informed consent. We asked everybody, starting from that Saturday, who came to get a vaccine, if you agree, please, before you get the vaccine, go do a serology test, sign, of course, an informed consent, and give us data on comorbidities, demographic data, and please agree to come initially on a weekly basis, but later on a monthly basis to do serology testing. And eventually we had around 9,000 healthcare workers who agreed, and we had this amazing follow-up. I should say already at this stage that later on, of course, this became much more complicated when we had the Omicron and later the XBB surges. And we ended formally the study actually just in the recent summer in June 2024. But after then, we still had a weekly, at the point of the Omicron, we let down, we understood that compliance is not that high anymore. But we still asked, especially during the Omicron surge, to do a weekly PCR. We had compliance with about 3,000 healthcare workers. And since XBB, and it's not ongoing, as I said, it's actually ended in June 2024, we still had a weekly symptom questionnaire that people would get on a text message, very simple, so they would answer. And that is the available data later on. So what did we do? Well, initially, to study the early vaccine effectiveness, we looked immediately, as I said, on 19th December, as healthcare workers started to get vaccinated, we looked at all of the healthcare workers and counted for each one the days, and we put them in a group, whether they were unvaccinated. So each healthcare worker would enter this cohort, and he could stay unvaccinated during this first month immediately after we started, or move once he got vaccinated to the next groups, whether it was the group of 1 to 14 days post first dose, and then so on. So at that early stage, actually just 1 month after vaccination started, we looked at the exposure days, number of exposure days for each group, the number of cases, and we could calculate the rate reduction for any PCR positive infection and for symptomatic COVID disease. And this was very early published in the Lancet. Next, we said, okay, we got second dose. We want to see what is the vaccine effectiveness of the fully vaccinated, what we thought then was fully vaccinated, the 2 doses. And this was the study that we carried out 3 months after vaccination was implemented. And again, looking at, on one hand, what was the patient days, we counted the patient days, but also we looked at the exposure days. And that allowed us to check, to assess not only the vaccine effectiveness for symptomatic infections and for all PCR, but also condition it on exposure and then really see what the VE for asymptomatic infections was and for infectivity. So we could also define how infected are these individuals who maybe even have an asymptomatic infection. And again, we showed that it was very highly effective, the second dose with these high numbers. Then when we get to the third dose, and we are now 5 months after the second dose, and we have around 12,000 healthcare workers vaccinated with the second dose, and we're starting to see emerging again infections. That was the Delta surge. And I'll tell you later how the third dose actually was approved in Israel, but at this point it was approved, and we started getting healthcare workers who at some point are getting a third dose. And again, in a similar way, we looked at the person days of people who had only 2 doses and compared it to those with the third dose. We didn't have, unfortunately, or luckily, anybody, or not anybody, but we had very few number of people who were unvaccinated, so we could not compare the third dose to those, but just comparing it to those who got a second dose 5 months previously. And again, we showed a very high vaccine effectiveness of the third dose during the surge. Next, when the Omicron surge started and emerged, still nobody thought at that stage of giving a fourth dose, but we started seeing this surge and people started talking, okay, should we give another vaccine dose? We said, if we do that, let's do it in a controlled trial. And at that stage, it was not approved yet. And we did an open label controlled intervention. And this time we wanted to see whether there was a difference between the Moderna vaccine and the Pfizer vaccine. I didn't say, but in Israel initially, the only vaccine available was the Pfizer vaccine. That also allowed us to do a lot of things more simply because we just had a single vaccine. There were no issues on that. So here we got also the Moderna vaccine, and we had one arm with Pfizer vaccine, one arm with Moderna, and we matched a control group from, again, these are all these healthcare workers. Some of them, at some point, the fourth dose was approved, and some of them actually were excluded eventually because they did get a fourth dose. And when we compare this, you can see here the cumulative incidence of infections, and you see that despite the vaccine, for some of them, still nearly 20% got infected. You see there is a slight difference. In a minute, I'll show you the results. But anyway, it was obviously not as good as we saw before for the previous surveys. And to summarize what we had on vaccine effectiveness, we showed the vaccine effectiveness of the first dose. And I think that was the impact. The most important was that it could support a delay in the second dose for countries who had lack of supply, and that was actually what was done in the UK. For the second dose, again, we showed that it's also reducing infectivity, which I think was really important. Third dose, we showed how important it is and the superiority over a second dose. And the fourth dose, we showed that there are only marginal benefits for, again, remember, we're talking about young, relatively young, healthy adults. And obviously, there are other works that show how important it is for vulnerable populations and for elderly. We also showed in a later follow-up study, which I didn't show here, but that this is really short-term effectiveness. It's only for about 13 weeks. So if you know there's 30, maybe there's use in getting the vaccine, it's only for the short term. So that was initially looking at vaccine effectiveness, but as I said, we had SARA, we had PBMCs, and we wanted to study the immune response both to infection, to vaccination, to booster doses, and maybe try to find correlates of protection. And our first study on correlates of protection was this paper very early on when we just finished giving the second dose. And I should say that within a month, about 90% of our healthcare workers got the second dose. It was really very rapid rollout of the vaccine. But early on, we already started seeing breakthrough infections. And at the point where we had 39 cases, we said, okay, let's try and understand why are some people, is there some kind of a correlate of protection that we can find? And among these 39 cases, 22 were included and signed an informed consent, and we had their serology, and we had a serology testing just before they got infected. So we said, let's match those two controls, and people who did not get infected, but had the same age, the same time since vaccination, all of that. And let's see if we can actually find a correlate of protection in among the antibodies that we have, among the humoral response. So we had 22 healthcare workers who had the infection compared with 104 who were uninfected. And what we saw is that indeed the cases had a lower neutralizing antibody in the, we call it peri-infection because for some it was during the first day of infection, but still it's actually, now I can say more, it's the pre-infection titers. So they had lower neutralizing antibodies, they have lower IgG, but this was significant despite the small number of cases and controls that we had here. Moreover, we saw that there was a correlation between the peri-infection neutralizing antibody titer, meaning the higher they had neutralizing titer, if they did get infected, then they were less infected, and that was according to the CT value. So the higher neutralizing antibody you have, if you are infected, you have a lower viral load, and that is also important regarding the vaccine. What about then later, and I'm jumping back and forth in time, but then when we got to the Omicron infections, we asked, is this also, are the antibodies also a correlate of protection? Because what we saw is that people got infected despite the fact that we were following them along, and we saw that they had antibodies, they were pretty high even. Still, people got infected, and it was, a huge surge. So, do antibodies still play a role? What's happening there? And what we did here is we followed and we looked at the nearly 4,000 healthcare workers where we had this good follow-up data, and those that received a third dose and participated in our study. Among them, some of them already got the fourth dose, some of them had some previous infections, and we looked just at those who were not, included all of them. It was nearly 3,000 healthcare workers. All of these, look at the numbers, 1,100 were infected with Omicron, it's amazing, and 1,700 were not infected. And during just these 2 initial months of the Omicron infections, and we compared the probability of getting infection according to your pre-infection antibody titers. And so what we see is, again, there is correlation, more so for neutralizing antibodies, but as you see, the numbers are higher. So we need a higher titer, and still, even with these were considered very high titers, over 2,000, still just about 10% had a probability of getting infection. So obviously it was less protective, but still neutralizing antibodies could and do play a role. Moreover, we looked whether they also play a role in the degree of how much will you get, how severe will the infection be. So during Omicron, and again, these are healthcare workers, we didn't have any severe cases, of course, but we did divide it to what we called a substantial symptomatic infection, meaning that these healthcare workers needed to stay in bed for at least 24 hours, had fever, and not just a mild sort of flu-like, very mild infection. And we see here again that, neutralizing antibodies definitely, and the humoral response plays a role even in the degree of how severe your infection would be. As I said, we were following all of these patients, so we could also really follow and look at the waning of the humoral immune response. And actually, this work, which was after 6 months of the second dose, together with our study on antibodies as a correlative protection, the one I showed initially, and another work showing that it's not just waning of immunity, but also waning of vaccines effectiveness, is what brought the decision for a third dose in Israel. And this was actually the first time that Israel decided, before FDA even approved it, to give a vaccine. That never happened before. And after Israel decided on a third dose, we had discussions with FDA and NIH telling them about our results, and then the decision went on and spread worldwide. What do we gain from repeated boosting? And this is a follow-up. What we see here in the pink is actually what I showed before after the second dose. And then if we compare it to the blue is the third dose, and then the purple is the fourth dose. And what we see is that, yes, there is a difference. The third dose definitely gives a different response. It's much higher, and it stays at the higher, the waning is much slower, and that a fourth dose doesn't really gain too much more than a third dose. Yeah, it's good if you need it, because if you're down here, it's good to be up here if there is infection, but it is pretty much short for about 13 weeks, as I said before, and you can see it also in this vaccine effectiveness, which is nice, but not as impressive as before, and also only for a short while. So to summarize, what were the impacts and implications for policy and practice of our works? I showed the early vaccine effectiveness, which supported the delay of the second dose where there's lack of supply. Antibodies, particularly neutralizing antibodies, are correlates of protection from infection of disease, but it was difficult to define threshold. We did other studies that I didn't show, but really difficult to sort of say, okay, that's the threshold like we have for hepatitis or for other infections. And a booster is required as part of probably the primary schedule. A second boost and then a third and a fourth and so on, they have low vaccine effectiveness. It's not just for Omicron, it's for repeated surges right now also, but they are effective for the short term, probably only marginal benefits for young, healthy healthcare workers, but as others have shown, it is, of course, important for other populations. Also, I didn't show, but we used our healthcare worker cohort as a control group for other patients in Sheba that we tested for immunosuppression patients, pregnant women, to really show the real-world evidence for populations that were not tested, obviously, in the clinical trials. And these are, again, just a summary of all of the broad achievements and topics that we were studying during this time. And luckily, at approximately this time, Dr. Danny Duick, who is the Chief of Human Immunology at the VRC, the Vaccine Research Center at NIH, was on a sabbatical in Israel. And he heard about the healthcare worker cohort and our huge bank of blood sample bank. And he said, wow, that could be really a source for monoclonal antibody discovery, because at that point, he was just about to publish a method that he developed, which he calls RATPIG, which is Rapid Assembly, Transfection, and Production of Immunoglobulins. And he likes that name, and that's how it's called. The idea is that actually, if you have PBMCs and you sort them and you know which probe and what you're looking for, you can discover monoclonals within 2 weeks, actually even 1 week, what typically takes 3 months or more, eventually about 6 months. So very rapidly, and he said, let's do this collaboration. And what we did is send Dr. Yaniv Lustig, who's head of the virology and part of our group. And we sent him to the NIH for half a year to study the method. And he took with him 26 samples, which were selected from our healthcare worker cohort. And they were selected according to their exposure by vaccine, by infection, all of that. And he produced over 2,500 monoclonal antibodies using the RATPIG method. And he got high affinity RBD binding monoclonals, and then looking for the broad neutralizing one. And eventually, we could define 2 monoclonals. One was from a patient, or not a patient, like the healthcare worker, who was exposed to Omicron, but never vaccinated. I said that was rare, but we did have one of those. And he did agree to give us the blood and get into the study. So he was one that had a very highly specific Omicron neutralizing monoclonal antibody, but it was very narrow. While another one that we proceeded with was a monoclonal that was very broadly neutralizing antibody of a healthcare worker who was vaccinated and also got infected. So this was sort of a proof of concept of this idea. And with that, this actually led to the idea that we should establish the Sheba Pandemic Preparedness Research Institute And together we partnered with the NIH, and the idea was to establish a multidisciplinary, multifaceted, collaborative research to translate basic science research into infectious diseases and into clinical products. And these biological countermeasures, actually, for us right now, what we're concentrated is these monoclonal antibodies will be rapidly deployable in the event of an epidemic and a pandemic threat. And the idea was to look at what we did with COVID, and let's take it as a proof of concept. So we had a lot of epi studies, we did immunological studies, then we discovered monoclonals, and the idea was let's go to the industry and then get it into clinical studies. And we said, okay, if we did this for COVID, let's think of what are the future pandemics that we should be thinking of. And of course, avian flu is one of them. We said, let's build a cohort of people who were exposed, and we had it a year before, a big outbreak of cranes in the north of Israel. So we went and established this small cohort of people who were exposed to these cranes. Similarly, we had the collaborative research with Palestinian colleagues, and they recruited people who went to the Hajj pilgrims and came back. And the idea was, let's see, what kind of infections they might be bringing with them. And there's, of course, in Saudi, the issue of MERS, and so on, a cohort of travelers, and so on. So as we were, together sitting and thinking of the 5-year plan ahead, first, of course, the SARS-CoV, let's look at further avian flu, as I said. Then we had the monkeypox outbreak. We said maybe we should build a cohort of people who were exposed to monkeypox, or just people who were prone to STDs. And at that point, each one of us sort of started bringing his older loves, or before what we call the pre-COVID interests in science. And for me, obviously, it was AMR, and thinking of a monoclonal for AMR was still there. It's very complicated. Bacteria are much more sophisticated, unfortunately. And then Yaniv Lustig, who's a virologist, said, what about West Nile? And I said, West Nile, it's not a big issue. It's obviously there's not going to be any pandemic of West Nile. Why do we need to? I was pretty much against this. And he said, but this would be a really good proof of concept, because we have all these blood samples. We don't need to build a new cohort. We can just take blood samples from our cohort, because in Israel, we knew that about 10% of Israelis are exposed to West Nile. So we expect it to be about 10%, so we can find it. And then let's just run it. It could be a simple way to say, well, it's not let's try it and go for that. And that's how we decided to go for West Nile. And I guess here I should give at least a little bit of background on what is West Nile and why do we care. So West Nile is a mosquito-borne infection. The reservoir is birds, and mainly crows, but others as well, and also migratory birds. And the mosquitoes get their blood from the bird, infect other birds. They're mostly not with any symptoms, although they can be. And then if they bite a human being or horses, then we get also infected. And what is the disease? Mostly it's asymptomatic infections, and that's why these 10% of people that we expect to find in Israel mostly are, they never knew they had any West Nile, yet 20% have what we call West Nile fever. West Nile fever is typically a rash, some myalgia, not a very severe infection, but there is, although it's only less than 1%, it's a very severe infection, which is neuroinvasive West Nile, and that is typically encephalitis, but not just. And these are typically healthy people who within a day, really have a very severe encephalitis with high mortality, and it's indeed a very severe disease. So how common is it and how do we treat it? In the United States, West Nile is the leading cause of US arboviral disease, but you say how frequent is that? So on an annual basis, there are about 800 to 10,000 cases differing from year to year. In Europe, also depending on the areas, in Italy, there were frequent outbreaks, in Greece, and all of this brought the New England to write this, to publish this, maybe it's time to revisit vaccinations because it is in many ways a neglected disease. There was just this year, a month ago or so, a paper in the Lancet showing that there is a correlation between high temperatures and they're associated with increased risk of West Nile neuroinvasive disease in different countries. A very nice paper. So how common is it? It's pretty much a neglected disease, but maybe with the global warming, it shouldn't be, and it might be something that we should think of. And regarding treatment, I didn't say, but there's no available treatment whatsoever. It's only supported. And once you see these patients with encephalitis, it's really horrifying. Okay, so we were at that stage and we decided, okay, proof of concept, let's go for this. And we got 300 sera from the sample, the bank, and indeed 11% were positive for IgG. Then we looked for neutralizing efficiency, and the correlation was not that great. We had some that had relatively low IgG, but they did have high neutralization, and we had others with high IgG, but no neutralizations. So we picked those who had high neutralizing antibodies and called them and asked them if they would be willing to give PBMCs. And we had, at that stage, about 20 or so that we had. And at that point, we, Danny Duick met us with Ted Pearson from the NIH, who's responsible for West Nile and arboviral infections. And he was very much enthusiastic about this idea. And we started, so they produced the probes, and then we established the cohort. And we were screening already for which would be the best candidate, doing the rat pick. And actually, at the point that we had, we were at the monoclonal antibody selection, we had 5 candidates. We were just the next week about to put it in the first mouse experiment. And at that stage, we have this huge outbreak in Israel of West Nile virus. And what do I mean by huge outbreak? I mean, this is 2024, 900 cases. We never saw anything like that before. The highest outbreak before was in 2000, 24 years ago. And if you look at each year, typically it starts only really in the summer, like July or even August. Here, it was end of May when we started seeing these cases. And that is, in a way, still an enigma. Why? What happened this year? Is it just exceptional warm spring and there were high temperatures during that spring? Is it the war? Because we were at war during that summer. And that meant that a lot of people in Israel were evacuated from the north, a lot from the south. So much more crowding. And probably birds as well were not in the, running away from all the missiles, and maybe they were all, so that was again, hypothesis, but, and of course, is there some uncontrolled mosquito source that we don't know of? Eventually there was at least one uncontrolled mosquito source, so that was part of it. But what other reasons were, we don't know, but we typically don't know, like what happened in 2000, and similarly in the United States, we have some years that are really high, others that are not as high. So that is sort of, we're not sure about what happened there. Anyway, back to our story at Sheba, we were somewhere here in the beginning of June, and just in our hospital, we had initially 2 cases within a week, and that was very strange. And then when we had 7 patients per week of West Nile, that's unheard of. We have typically, 2 patients this season or something, because we have 30 hospitals in Israel. So how common would that be? We understood something bad is happening. And we said, what do we do? And again, we see these patients, most of them were around, the median was around 70 years old, but relatively healthy. One was a CEO in this company. Another one was this 70-year-old who was just traveling to Nepal hiking and coming. So these were really typically healthy adults coming in and just getting encephalitis. So it was really scary. We said, what do we do? No treatments whatsoever. And we, of course, thought, oh, if we only have our monoclonal, what a clinical trial can we do? But we didn't have it. So we said, okay, we know that, or we don't know, but we think that these monoclonals would be helpful. We know that they were taken from healthcare workers who had these. Maybe if we can get plasma from these healthcare workers with high neutralizing titers, and we give that, maybe that could help. And we decided to run it as a randomized, double-blind clinical trial. We thought that that's stage that, if we have 20, 30 patients every week now for another few weeks, we can probably recruit 100 by the end of the season. And with that, we can definitely look at some outcomes that we can find something which would be significant. So we offered also other hospitals to join, and we thought that we would do a multi-center study. Apparently that didn't happen because it took time for each hospital to run its IRB and all of that. But at Sheba, we did get it very rapidly. And we actually sent the first letter to healthcare workers to see if we can recruit people to give the plasma on June 27th. And 1 week later, the first patient was already recruited. So what did we do? We had over 2,000 healthcare workers who tested for IgG, and 12% were positive among these. All of these were tested for neutralizing antibodies, and only those that had neutralizing antibody titers, we wanted to have it as high as possible, but initially we couldn't be that particular. And whoever had, we asked them to come and give plasma. Giving plasma means sitting there for 2 hours, giving your blood. It's complicated. This is where the CEO of Sheba, Professor Price, was luckily also with neutralizing antibodies, so he could, and he was very happy to volunteer. So the inclusion criteria for donating plasma, that is also complicated, is either men or women before ever being pregnant, because apparently after pregnancy we have all kinds of antibodies and we're not allowed to donate plasma. Yes, blood, but not plasma. And we wanted high neutralizing plasma, but we couldn't be that particular initially. Later on, we could, and then we asked only for those with higher doses. And then what we did is, among the patients that entered the hospital at that time, and again, in the first few weeks, it was around 20 patients, we screened them, and of course, we offered them to join this trial. And we randomized them, whoever agreed, to plasma or saline, 2 to 1. So we would give more plasma. As you can see the difference. So we had to put it in a black bag, so it would really be blinded. And this is what we. This is the study schedule. So initially we screened, and the inclusion criteria were if it was a hospitalized West Nile patient with the age of over 60 or lower if he was immunocompromised, and that the diagnosis was within 72 hours. But this is just a diagnosis. The symptoms eventually, and we understood we can't put that as a criteria, but as you see, most of them arrived to the hospital after at least 7 days of already having some symptoms. And the symptoms, we were not, we didn't put only neurological signs because we thought maybe that's too late. So we said anybody who is symptomatic, but symptomatic had to be at least fever or a neurological symptom. If you just had rash, that was not enough. But these typically don't go into the hospital also. On first day, we gave them either the plasma or placebo, and we assessed neurologic and cognitive and functional assessment. And we looked at the viral load, both in the blood and the urine. Then we had a follow-up on days 3, 5, 7, and 14. Eventually at the end of study, which was day 30 and then day 90, we looked at the outcomes. And we decided that the primary outcome is unfavorable outcome, meaning either mortality or a functional deterioration. And the secondary outcomes would be, of course, also on day 90, looking also at mortality, and each one separately, neurologic, cognitive, functional, and also a composite outcome of all of these. The statistical plan was to run a logistic regression model and to adjust for immunosuppression and the time from symptom onset to treatment. And our exposure, meaning the neutralizing antibody titers, was modeled in 3 ways: either binary if you got neutralizing antibodies or not, or categorically if you had high neutralizing or low or placebo, or continuously looking at the log of the neutralizing antibodies and the outcomes I just mentioned. So this is the diagram of who was eventually randomized. So we didn't get, unfortunately, to 100 patients. Only 38 were recruited. And by the time we finished that, pretty much the outbreak was over. We also, 4 were excluded because either there was no B-type or AB-type plasma, which is much more rare. And again, at later stages, that was not an issue, but initially it was. And 1 who eventually had a West Nile diagnosis 2 months earlier. So we had 34 patients, and we thought, what are the chances we're going to see anything? But this, in a minute, I'll show you what we did see. Anyway, they were randomized, and 12 got saline, 22 got neutralizing plasma. And as you can see here, again, the neutralizing plasma, we always picked whatever the highest we had for that blood type. And eventually, we had the neutralizing titers that the patients got, as you can see here. So we also divided it to what we called high neutralizing. Is this really high or no? I'm not sure. It was in a way arbitrary, or looking at the median. And then what was low, and then of course those who received saline did not get any neutralizing antibodies. So just again, the study population, the mean age, as I said, was over 70, 73 or 79. And we had we had pretty much similar male and female actually. We had slightly more B-cell immune deficient in the neutralizing plasma group. And other than that, neuroinvasive, that's probably important. So I said that time from initial symptom was about a week, okay, for both of the groups. And among these patients, about 75 to 77% had neuroinvasive disease. The others just had fever. And this is on recruitment. Okay, so what are the results? First of all, safety, of course, if it's a study with this kind of a drug, we need to look at that. We had a single case of allergic reactions, which is something that we see when we give plasma. And of course, we had SAEs because all were not related to the plasma, or to the treatment, but to the disease, because it was just disease deterioration. We had several cases of death. So what is our primary outcome? We said it's a composite outcome of 30-day mortality and neurologic deterioration. And we define neurologic deterioration as decrease in the Bartel index. Bartel index is one way that you can assess what we call ADL, which is activities of daily living. And so what we see is nothing, doesn't look good. But when we look at how many of these actually died, because remember, all of the dead cases are also included here, actually, we see something very different. And so we looked at the secondary outcome, which is mortality, separately, and indeed we see that it was very significant, and more so when we divided to the low titer and the high titer. The group, again, these are small groups, but none of the people with the high titer died, while people in the low titer did, and people in the saline even more so. When we look and divide it to stratified it by immunocompromised status initially, we see again that the plasma probably affected even much more, and altogether the mortality, and probably I'll show it here best, is these are the final results of the logistic regression, and this is adjusted for the immunosuppression in the time from symptom. And here we look at the exposure modeled as a continuous variable, meaning that we look at the change for each log of neutralizing titer that is higher. So actually the relative risk was about 20% lower for each neutralizing titer that we could go, log of neutralizing titer that we go higher, which is very significant, and mainly for this such a small group. And also the Glasgow Coma Scale, which is the consciousness of the patient. But when we looked at Mini-Mental, which is a measure of cognitive at 30 days or 90 days, or as I said before, the Bartel, which is the ADL, we didn't see anything significant. And that's why, of course, the composite did not come out significant. The limitations are obviously the very small sample size and maybe a longer follow-up if needed. This is by 90 days, we see that all of the patients, or 50% of the patients, really still had bad neurologic deterioration. Does this improve later on is a question. And some of the outcomes maybe are not subtle enough. Maybe we need to look at more than just, what is neurologic deterioration? That's another question. And of course, this is a very complicated study. As you can understand, the logistics are really very, very complex. And obviously, I don't think it's really generalizable to start running trials like this if you have an outbreak. But we think that this might be a proof of concept for the idea of the monoclonal antibodies. So to conclude this part, neutralizing plasma given to the patients with West Nile, and again, mostly they were neuroinvasive within 72 hours of diagnosis, not of the symptoms. And so, within 7 days from the onset of symptoms, was associated with a significant decreased risk of mortality. And we observed a dose response effect. So the higher neutralizing, the more protective it is. And yet 50% of both groups had functional deterioration at 90 days. And this maybe could suggest that if we would be able to treat them earlier, maybe they would not, we would be protected against that as well. Maybe giving it 7 days is just too late for that effect. But anyway, we think that this suggests that neutralizing monoclonal antibodies could be used for treatment and prevention. And together, just to summarize up everything, so the next steps that we are now toward a West Nile monoclonal is, one, we think that this plasma trial that we had is in a way a proof of concept. And we have right now 5 leading West Nile monoclonal antibody candidates that actually already finished a successful mouse experiment. And currently we are in the 2nd week of the developability studies of 2 of them at the NIH. And we're now negotiating with several CROs for production. And we hope to succeed and have a product ready for a phase 1 or 2A, maybe in the summer, not this coming summer, but maybe the summer after that, and actually really do the whole story from the beginning, getting it from patients until giving a product back to in a clinical study to our patients. So with that, I'd like to finish and be happy to take questions and just acknowledge a lot of groups here are involved. First of all, really a lot of thanks to Danny Duick and his team at the NIH, and Ted Pearson and his team at the NIH, Yael Tolengi, who is the lab manager of the SPRI lab. And Noam, Jan, and Michal were responsible for many of the studies on the COVID. And then the rest of the group, the virology, I mentioned Yaniv Lustig. For the West Nile, we had a collaboration also with a neurologist who did the assessments from the Sheba Blood Bank and from the Central Blood Bank of Israel to get all these plasmas. And the ID unit, the microbiology, and of course the Sheba management, and mainly Professor Price, who really gave support to everything budget-wise and not budget-wise, and so on. And currently, we got nice funding from the Helmsley Foundation. So obviously, I need to thank them. And that's it.
Thank you so much. We have some time for questions now, and if you're on Zoom, feel free to put them in the chat too. I'll start. I won't throw this.
Okay. Thank you so much. This was so cool. I was wondering about the West Nile clinical study, if you have any of the data on looking at mortality as a time to event outcome instead of a cumulative outcome, and what the distribution of times to death is, and whether you have some like mechanistic hypothesis of if the antibodies provide like supportive care until people can mount their own immune response, and so you're sort of like pushing off the time, the mortality. I just wonder if that gets around some of the issues with the follow-up.
Yeah, so the death events were all, and that's why it was similar 30 and 90, because actually we have 2 cases and we're still following. It's not part of the study, but we were of course interested in everybody who went through the study. So there were 2 further deaths after of the group that received the saline. And but most of them were really dying relatively soon. It was like within 2 weeks. And they deteriorated neurologically very fast, and most of the deaths were among the immune compromised patients. Yeah, behind you.
Thank you so much for that talk. It was really incredible work that you did so quickly during the pandemic. So I was a resident during the COVID pandemic at Hopkins, and there were big trials looking at convalescent plasma for treatment of COVID that ended up being ineffective, and we now no longer use it outside of like very case by case kind of situations where people are very immunocompromised. So I'm wondering if you could comment on what about the way that you collected the serum for the West Nile study, and or something about the immunology of West Nile versus COVID, like this being effective.
Yeah, I think for COVID as well, probably, and I think there's at least 2 studies on IVIG for West Nile, which were not successful also. And I assume that also if people would just give convalescent plasma, it would not necessarily be, you really need to check if you have neutralizing, and you see even those that had low neutralizing antibodies, they were less effective. We really could see it. So you really need, probably if you would take from COVID, and I knew we would go in Sheba and say, oh, I'm 200, oh, I'm 2000. Everybody knew how much antibodies they have. So I can tell you that my antibodies were typically not very high. So, I got all the vaccines. I was at probably if I would donate that plasma, maybe it wouldn't. So I think you really need to know. And that's why these kind of things are not necessarily feasible. But monoclonal antibodies for COVID are, of course, efficient. Yeah, to the back.
Thanks. Questions. I was curious if you could reflect a little bit, just kind of on the process and the logistics for setting up all of these studies so quickly. What did you learn along the way? What were things that really bogged you down? What kind of decisions did you have to make, et cetera?
Yeah, so that's a very good question, and it was really, I think, the fact that we did it so fast was the success of this. And the reason we succeeded in this was, one, I think we really had very large support from the management of Sheba. Without this, it would not go. And once they said, this is really important, then they got the IRB to sit and have an urgent meeting and agree, all of these things were really, once the management is there. And of course, I have to say, we really have a good group. So, my people were really all devoted. We were 24/7 working on this, while everybody was just staying at home. And so we felt like, we have a mission, we can do something. So, yeah,
Thanks.
but it was very complicated, yeah. And it's funny just to say, because when we started with this West Nile, and it was like 2 years after sort of things were coming down, we said, oh, this feels like COVID again, we're all, again, okay, we have to run a clinical study within, 2 weeks. It was doing crazy things. And, but once you sort of get everybody into it, then, yeah, it worked.
You mentioned that you weren't sure about the cause of the West Nile epidemic that you guys had, that maybe it was an uncontrolled mosquito source. I was just kind of curious, like you said, maybe there was one. What was the story behind that?
Yeah, so there was initially, all of the people, so it was also, something which required collaboration of the agriculture department, the health department, and lots of departments were working there together, and that is typically exceptional. It doesn't happen, but it had to happen then, and it did. And each one was saying, no, nothing. All of the mosquitoes are taken care of as usual, nothing happened. But then they did find one specific place, which was near Tel Aviv, and that was a source. It was all urban, a lot of it was urban, and that was not treated. There was just a single source of, I don't know how you call it. It's not wastewater, but something that you take care of water. And there was really one single source. And after they did take care of it, again, was it this or again, the weather changing? It all happened together. So it's difficult to say. But in Israel, you would hear some that would say, oh, obviously it's all this source. And others that would say there is a single paper out by now. And they're saying it's because of the warm weather in the spring. So I think it's probably a mixture of them all.
Thank you.
Okay, yeah, we are out of time now. That was a very interesting talk. Thank you again so much.